Start, madam.
Okay, the topic that we shall be dealing is about the pneumococcus. As in the last classes, we have studied about staphylococcus and the streptococcus, which are gram-positive organism. This organism is also a gram-positive organism which exhibits a lanceolated appearance shape, that is flame-shaped appearance, and sometimes this organism can also appear in the form of a cocci or a spherical resembling the streptococcus. And this organism was initially classified as Diplococcus pneumonia, but later it is renamed as Streptococcus pneumonia because of the genetic similarity with the Streptococcus species. But it has its own characteristic which makes it different from the Streptococcus species. The difference is in its morphology, the biosolubility, optogen sensitivity, and the expression of a polysaccharide capsule that is the most important component that exhibits the virulence of this particular organism. And this pneumococcus is a normal inhabitant as a normal flora of the upper respiratory tract, but mostly associated with the pathogenesis or the disease of pneumonia or the otitis media. And this organism also have the ability to cause other infections such as sinusitis, bronchitis, bacteremia, and meningitis when the host resistance usually gets lowered. Now coming to the history, this particular organism, that is the pneumococcus, was discovered by Pasteur and Sternberg in 1881 independently, and the relationship of this organism with the pneumonia was established by two scientists, Frenkel and Welsh-Bowman, in 1886. That is, the pathogenesis of this organism is usually associated with the polysaccharide capsule. So this was demonstrated by Frenkel and Welsh-Bowman in 1886. Coming to the morphology of this organism, these organisms are very tiny and they show typically small cell appearance and measuring about 1 micrometer in diameter and slightly elongated cocci. That is, these are not completely spherical in nature, but they have one end which is pointed, whereas the other end which is a broad one. So they are appearing in the form of a lanceolated shape or a flame-shaped appearance. And this organism appears in pairs and hence called as diplococci, and these diplococci usually are in adjacent to each other on their broad shaped spherical ends, and the organism exhibits capsulated material around its surface, which is the one that is related to the virulence of this organism, and it is this particular feature that makes it different from the other group of organisms. And the capsular material can be lost when on a serial subculture, and the organism might appear spherical in nature due to the loss of its morphology. And this organism is non-motile and non-sporing in nature. The capsular material can be demonstrated with the help of different special staining techniques that helps to identify this organism in the cultural media. Coming to the cultural characteristics, how these organisms exhibits their characteristics on a particular culture or a media, that is the nutrition which is provided to the microorganism. These organisms cannot be easily grown on a natural or a normal media, but they have to be provided with special ingredients to isolate these organisms. Such type of media is called as the enriched media. So enriched media is the one where we enrich the media by adding special ingredients such as blood, serum or egg, etc. For this organism, we usually isolate it in the blood agar, and the characteristics of this organism is they are aerobes and facultative anaerobes. That is, in presence or absence of oxygen, these organisms have the ability to survive. And the temperature, the optimum temperature for the growth of this organism is 37 degrees centigrade, ranging from 20 to 40. So that is the optimum temperature where you can see the high growth of this organism is around 37 degrees centigrade. The pH they grow is around 7 to 8 or 7.8 is the optimum pH where you can isolate this organism in the media. Now coming to the selective media for the growth of this organism, the enriched media that is used is a blood agar, and on the blood agar initially after growing for 24 hours they show characteristic appearance of they look like they are small, dome shaped, they are glistening, showing alpha hemolysis, which is similar to the Streptococcus viridans, which shows partial hemolysis or the greenish discoloration on the the media. On further incubation, these organisms or the colonies looks like flat, where the edges are slightly raised and they show a central ambulation. That is, we can see beautiful peripheral rings on the when viewed from the top, similar to that of a carom coin or a Drottman's colonies. And the capsular material in most of the organism, that is type 3 strains of pneumococci, exhibit more amount of capsular material, and hence these organisms can be isolated in the form of a mucoid colonies. That is, the more capsular material produced by this organism can be easily identified by the mucoid colonies on the media. Now, on incubation in anaerobic condition, because they are facultative anaerobes, these organisms shows complete lysis around the zone, which is called as a beta hemolysis, and it is because of a specific protein that is released by the organism, which is called as the hemolysin O, which is streptolysin O toxin similar that can be seen in the Streptococcus species. So, it is a heat labile hemolysin O that brings about the beta hemolysis on the blood agar media. And in liquid media, this organism can be grown in a glucose broth, and the growth can be seen in the form of a turbidity. And this organism exhibits a property which is called as autolysis. That is, they can degrade or cause a lysis of themselves because of certain enzymes that are present in the organism that causes the autolysis. And this autolytic property can be easily demonstrated in the laboratory condition to isolate this organism. And the autolysis can be enhanced by bile solubility or sodium lauryl sulfate or any surface acting agent. And we should know that this autolysis is lost in the strain which are usually heat resistant one. Now, coming to the biochemical reactions, this biochemical reaction helps in identification based upon the biochemical properties that are brought about in the cultural media. These organisms can ferment many sugars producing acid but no gas. The most important fermentation that can be seen here is in the His serum water or serum slope, where the organism can bring about the fermentation, and they are easily detectable on this particular media. And the most important property of this organism that distinguishes it from the other organism is the inulin fermentation. It is the only organism that has the ability to ferment inulin, and it is a characteristic feature that helps to identify the organism in the biochemical nature. Now, coming to the bile solubility, these organisms are bile soluble. They get easily lysed when exposed to the bile. That is, this can be easily identified when we add 1 drop of 10% bile or sodium deoxycholate to the media or the broth containing the actively growing microorganism. You can see a clear solution that is formed due to the lysis of the organism when it is exposed to the bile. So, this is the characteristic property of the pneumococcus which is used for the diagnostic purpose. And this particular test can also be carried out on a blood agar media when a drop of this 10% sodium deoxycholate is added to the organism which are present on the blood agar. They causes the lysis and shows a clearing of the zone. So, these are the two methods by which we can identify that the organism is usually bile soluble. And the property of autolysis, as said, is because of the presence of an enzyme which is called as autolytic amidase that has the ability to cause the autolysis of the organism. And this amidase acts on the most important component of the organism, that is the peptidoglycan layer. It breaks the bond between the alanine and the muramic acid, thereby causing the lysis of the organism. Apart from this, this organism is catalase and oxidase negative. That is, you cannot see a positive reaction of the catalase and oxidase, which is only the characteristic feature of the Neisseria. So, this organism, how do we identify is with the help of fermentation of the sugars, inulin fermentation, and the bile solubility test. These are the three characteristic features by which you can identify the organism in the cultural media. Now, coming to the resistance of the organism, as we know, resistance of the organism is very important for us to go about with the treatment of that particular organism. We should know whether the organism is a sensitive organism or a resistant organism towards different types of antibiotics. So, once we know the characteristic feature, we can go ahead with the treatment with that particular type of chemotherapy. So, coming to the resistance, these organisms are highly delicate in nature, and the thermal dead point, they can be easily denatured with the heat at 52 degrees centigrade for when exposed to 15 minutes, and they bring about the autolysis because of the autolytic amidase that is present. And on incubation of this organism for a longer time, they usually die because of the accumulation of the toxic peroxidase. And this organism can be easily saved or preserved for a longer period of time by a process which is called as a lyophilization or defreezing. And these organism exhibit sensitivity to different types of antibiotics. But recently we have found that there are certain species of pneumococcus that have developed resistance, and hence we have to go for an alternative treatment with the other type of antibiotic. Initially these organisms were highly sensitive to penicillin, and the beta-lactam ring was usually effective on the organism. But because the organism were able to gain resistance against this particular antibiotic, they are able to grow more in number and cause the infection. And the resistance in this particular group of organism is not because of the production of beta-lactamase, which is exhibited by the other organism, but these organisms are so smart that they are able to change their penicillin binding protein that is present on its surface. Thereby the penicillin is made inactive by the organism and is no longer able to cause the lysis of the organism, and hence most of the organisms have developed resistance towards the penicillin. And the most important characteristic feature is the change of the penicillin binding protein. Due to this change in the penicillin binding protein, they are not only resistant towards penicillin but towards different types of beta-lactam antibiotics. That's why there are certain species of organisms that have developed multi-drug resistance against different types of beta-lactam antibiotics, that is the penicillin G, penicillin V, etc. And recently a new type of organism has been discovered, which is called as drug-resistant Streptococcus pneumonia, which was recently found in the Sweden and is able to cause most of the infection worldwide. And the ability to be sensitive towards the optogen is another important characteristic feature that helps in identification of the organism. Optogen sensitivity. Optogen is ethyl hydrocuprin. When the organisms are inoculated onto the blood agar plate, and the filter paper disc that has been impregnated with the optogen, when it is placed and incubated, you can see a clearing of the zone which indicates the sensitivity of the organism towards the optogen. So this is another important characteristic feature with which we can identify the pneumococcus. The first is we can identify with the help of Boyle solubility. Next is the capsular material that is present in the organism. Third is the inulin fermentation, and the fourth is the optogen sensitivity. These are the four characteristic features that is exhibited exclusively by the pneumococci. Now coming to the antigenic property, antigens are nothing but that induces the production of antibodies, and the one that goes and interacts with the immune system. So here the antigens are the one that are highly virulent in nature and the reasons for the pathogenesis or the causation of the diseases. So here in the antigenic property, the specific feature that is exhibited is the type specific capsular material that is present around the organism, and this capsular material is easily able to diffuse into the exudates or into the media, and hence it is called as a specific soluble substances. And based upon this capsular material, you can classify the organism upon the antigenic nature of the capsula, that is in which way it is able to cause the infection. So in the lobar pneumonia, the capsular material that was isolated was typed into four different types. One is called as a type 1, type 2, type 3 and type 4. Later on, type 4 was again classified to many different types of serotypes. Nearly about 90 serotypes we know about this particular organism that are involved in the disease causation. Coming to the typing, how do we type this organism? It is mostly carried out with the help of serological technique, that is the agglutination reaction. Agglutination with the type specific sera and the capsular material helps to identify which type of capsular material and what is the serotype to which it belongs. Next is we can go with the precipitation test, where the specific soluble substance is treated with the serum of the patient to identify whether there is an interaction and the precipitation is positive or not, and based on that you can type the capsular material. And the third is the most important one, is a Quellung reaction, that is a capsular swelling reaction, also called as a Quellung reaction, which was discovered by Neufeld. And here in this particular test, what we do is we mix a drop of the capsular material, the organism which is active with the anti-sera with a few drops of methylene blue. And here we can see there is a swelling in the capsular material that can be easily differentiated into the media. And the capsular material is swollen and it is delineated and refractile in nature. So based on this particular characteristic, you can identify the pneumococcal capsular material. So these are the three methods by which we can go ahead with identification of the type specific organism that is involved in the pathogenesis. The other tests that are usually carried out is with the help of a species specific nucleoprotein, which is deeply seated inside the organism, and a somatic C antigen, which helps us to know the type specific organisms to which it usually belongs. Apart from this, during this particular type of infection, that is a pneumonia, a specific protein is produced inside the human body which is called as a beta globulin. It is the one that is able to agglutinate the somatic C antigen, and this is also given another name which is called as a C-reactive protein. And this C-reactive protein is not an antibody, but it is an acute phase protein produced by the hematocytes or hepatocytes during any bacterial infection. So apart from pneumococcal infection, similar type of C-reactive protein can also be seen in other bacterial infections, inflammatory responses, malignancies, and tissue destructions. So the index of CRP helps us to know to what range that particular patient is suffering from that infection. So the CRP can be taken as an index in the identification of the treatment that is going on with the rheumatic fever and other type of bacterial infections. To identify the C-reactive protein, we can go with the latex passive agglutination test, where the latex particles are usually coated with the anti-CRP antibody, and this helps to identify the CRP protein which is associated with the pneumonia. Now coming to the variation, this organism is very interesting in changing its physical or morphological characteristics from a smooth colonies to the rough colonies. So here there is a variation from the smooth colonies to the rough colonies on a media. The smooth colonies are usually produced by the organisms which are capsulated or which have capsule around themselves, whereas the rough colonies are the mutant types that have lost the ability to produce the capsules. So the characteristic feature of the R colonies is they are rough, they are non-capsulated, avirulent in nature, and auto-agglutinable. And these are the mutant types that are usually generated from the wild type, that is the S type of colonies. And this was this particular experiment that was carried out by Griffith, called as Griffith transforming principle, that has helped us to know that DNA acts as a genetic material, and moreover it also helped us to know the genetic exchange by transformation. So the rough colonies are able to produce the smooth colonies when they are introduced with the DNA of a particular serotype. The way in which they produce the colony depends upon the DNA of that particular serotype. So this is a transforming principle. The DNA is a transforming principle, and it is the one that was usually that carries the hereditary features from one generation to the other generation. So with the help of this particular experiment, that is the Griffith transforming principle, we know that DNA acts as a genetic material, and next important is the genetic exchange can also be brought about with the help of transformation. So the variation helps us to know that the organism is able to change from one wild type to the mutant type, that is the smooth colonies to the rough colonies, and the rough colonies can also be reverted back to the smooth colonies when they are inoculated or introduced with the DNA of a specific organism. Now coming to the other important virulence or the pathogenic characteristics of this organism, they exhibit toxins and other virulence factors, the most prominent being the hemolysins and the leukocidins. Hemolysins are the one that causes the lysis of the RBCs, and the leukocidins causes the destruction of the leukocytes. But because they show weak affinity towards the RBCs and leukocidins, the other two important are usually taken as the one to identify the pathogenesis of this particular infection. So as already discussed, the capsular material, which is a polysaccharide, is the one that helps to enhance the virulence in the host, that is causation of the disease in the host. And it is because of this particular capsular material that is the organism is able to escape the phagocytosis of the human immune response. And towards this particular capsular material, the human host responds by producing the antibodies or the neutralizing antibodies that go and interacts with the capsule. It is not easy to phagocytize the capsule in when it is in a fluid condition, but when it has got fixed to certain fibrin clots or epithelial cell, it is easy for its phagocytosis. This particular phenomena is called as a surface phagocytosis. So when it is free in solution, it can easily escape the phagocytosis and cause the infection. But when it is attached to certain firm substances, that can easily be phagocytized and can be eliminated from the human host without causing the infection. Apart from this, the organism is also able to produce a toxin which is called as a pneumolysin, and it is a membrane damaging protein that can bring about the cytotoxic activity or the complement activating in the human host. So complement systems are nothing but they are serum proteins that can eliminate the organism by forming a membrane attack complex. But during the activation of the complement, there is a large inflammatory response that is formed in the human host that can be deleterious to the human host and allowing the secondary infections to take place. Now, apart from this, the pneumococcal autolysin can damage the tissue by causing release of the content into the host cell and ultimately causing the damage to the host cell. Now coming to the pathogenicity, how this organism establishes itself and causes the infection in the human. Pneumococci, as already discussed, they are the normal inhabitants of the human nasorespiratory tract or the nasopharyngeal tract, and the organism can establish very well on the nasopharynx and can spread to the other nearby areas, causing the infections of middle ear, which is otitis media, and they can cause the infection in the paranasal sinuses or the respiratory tract. And the organism can cause meningitis when they enter into the blood, usually in the form of a bacteremia or in contiguity. That is, serially when they cause the infection of a particular organ, immediately they cause the infection of the nearby organ. So that is a contiguity where they can reach the meninges also and cause the meningitis. And apart from this, this organism can be very well seen in the blood in the early stages of the diseases, and hence it is called as a pneumococcal bacteremia. And this infection can either be endogenous or it can be exogenous. When a large amount of virulent strain usually infects the host, it is called as an exogenous strain, and the type 3 are mostly involved with this exogenous pathogenesis of the human host. Now coming to the pneumococcal infections, what are the symptoms and how can we identify these pneumococcal diseases is the first thing is otitis media, which is caused in the children's, sinusitis, and apart from this the most important being the pneumonia, and these are the secondary infections you can say that are caused by this organism. And the serotypes that are associated with otitis media and sinusitis are the 6, 14, 19F and 23F, and from the it also have the ability to cause the lobar and the bronchopneumonia, leading to acute tracheobronchitis and empyema. In case of lobar pneumonia, this organism can be easily aspirated from the nasopharyngeal secretion during the sleep or during the daytime. Now, in order to establish infection, it has to overcome the natural innate immunity. The natural innate immunity that usually expels these organisms is the cilia and the cuff reflex. So, if the organism is able to carry out these two properties properly, the organism can be eliminated without causing the disease inside the host. But once the organism is able to establish, then it can lead to cause of the pneumonia, and this can happen only when there is a lowering of the immune response in the host, or you can say that when there is an immune compromised patient where the immune system is not acting very well. In those cases, you can see the organism getting very well established and causing the infection. So the pathogenesis, that is the spread of the infection, is the establishment of the organism in the nasopharynx. Once they overcome the innate immune response, the organism multiply, and through the bronchial mucosa they move towards the lungs via the bronchial mucosa and the lymphatics, where they are able to establish themselves and cause the infection, that is pneumonia. And here you can identify the organism in the initial stages in the form of bacteremia, and apart from that, due to the diffusion of the capsular material, it can lead to toxemia. And in the adults, the strains that are mostly responsible for the pneumonia are 1 to 8 type, and in the children it is 6, 14, 19 and 23. And apart from that, the other infections that are caused by pneumonia are bronchopneumonia, which is a characteristic feature of this organism, that it is able to cause a secondary infection. Now, what does a secondary infection mean? Is when the immune system of the patient has come down due to the primary infections by the viruses or the other malignancies, these organisms are able to invade, that is the pneumococci are able to invade and cause the secondary infections. So this usually is caused in the patients that are immunocompromised patients, and hence it is called as a secondary infection. The reason for the secondary infection is already there is a damage to the respiratory epithelium and a continuous secretion of the bronchial mucosa is seen. This helps the organism to get attracted and invade the person and cause the pneumonia. So this bronchial pneumonia is a secondary infection. This sometimes can lead to chronic bronchitis where again the continuous secretion of the nasal discharge can attract the organism and cause the chronic bronchitis. The other reason for the chronic bronchitis can be the other organism, which is a Haemophilus influenza. Now coming to the meningitis, which is most serious cases that is associated with pneumococcus. If not treated in the primary stage, it can lead to meningitis, that is the inflammation of the meninges. So it is again a secondary response of this organism once the pneumonia has been established and cause otitis, sinusitis, conjunctivitis and lead to fatality of the that is a person. Nearly 20 to 30% usually die because of this meningitis. Now coming to the other associated pathogenesis of this organism is they are able to cause suppurative lesions such as empyema, pericarditis, otitis, sinusitis, pharyngitis. We have suppurative arthritis and peritonitis. Coming to the epidemiology, this organism is able to spread in a close community, and the natural infection is seen mostly in the animals, and in the humans they are usually carried in the person. So, an epidemiological characteristic feature of this organism is this organism is the one that causes natural infections in the animals, but in human it is rare, and the human which carries this organism in the nasopharynx are the carriers which can transmit the infection from person to person during cough reflexes, and the transmission is mostly via the droplet of nuclei or the fingers or the inhalation, and mostly these organisms are present in the throat, and hence the cough reflexes are said to be the one that can transmit the infection. And dissemination is in the crowding, where there is a heavy crowding. You can see most of the people suffering from this particular infection, and the person who carries these are called as the carriers, and they are called as the pharyngeal carriers because they carry the organism in the pharynx. And most in the case of the person who have got the lowered host resistance, you can see the organism causing infection in them. So if the immunity is good, then you can overcome this particular infection. But once the immunity comes down, then you are more prone to the infection with the pneumonia, mostly seen in the children's. And the person who has splenectomy or sickle cell diseases where the immune system has completely come down, in those type of people, pneumonia is very common and very recurrent. And the type of strain that is usually associated with the pneumonia is a virulent type 3 strain of this organism, and the lobar pneumonia being sporadic can also cause epidemic in a close community such as army camps, etc. So in these particular areas, if there is already an infection with the influenza or the other organism, you can see that the person being suffering with the pneumonia, that is with the pneumococcus. And the seasons where you can see the epidemics is during the winter, and the ages to which the organism causes the infection is the extreme ages, that is they can cause the disease in the infants or in the old people. Usually the adults are have a strong immune response, and hence they can overcome this particular disease. Now, how do you identify this organism in the lab? That is lab diagnosis. The lab diagnosis is very important to establish the positive agent so that we can go for the treatment. Now, because pneumonia is caused by different types of organism, there are more chances that there might cause a confusion in the diagnosis. So, hence you should have a good knowledge about the characteristic features of this organism so that you can identify and isolate them in the media and go for the antibiotic sensitivity testing and go for the treatment. So in the acute lobar pneumonia, the specimen that is collected is usually a rusty sputum, which is the characteristic feature of this organism. Go for the gram staining, identify the organism, and how do you identify is In the gram staining, they are gram-positive, lanceolated shape organism that can be seen in the smear. Next test that you can go ahead is a Quallang test, which is a characteristic feature of this organism. Again, mix the organism with the type-specific antisera with a few drops of methylene blue and the capsule swell, which indicates the presence of the capsular material, that is Pneumococcus. And when the sputum is collected, it has to be properly homogenized and then inoculated onto the blood media or the blood agar. 24 hours of inoculation is done with 5 to 10% carbon dioxide, so to enhance the growth of this organism. And in infants who cannot give the sputum voluntarily, you can go with the laryngeal swabs that have been coated with the antibody. Now in the respiratory secretions, inoculate the respiratory secretion onto the blood agar containing a gentamicin, which is made in a proportion of 5 microgram per ml. Now in the acute pneumonia, the organism have to be inoculated onto the blood agar along with the glucose broth incubated for the growth of the organism. And in acute otitis media, it is the aspiration of the fluid from the ear that helps to identify the organism. And in meningitis, you can identify the organism via the gram staining of the cerebrospinal fluid, or you can go with the culture, that is inoculate the cerebrospinal fluid onto the culture for the growth of the organism. If all these conditions are not possible, then we can go ahead with the serological testing, that is with the help of agglutination, precipitation, or more sensitive testing such as ELISA, RIA, or immunofluorescence, you can identify the organism. So here in the serological testing, we are going to identify the antibodies that are produced against the organism's antigenic characters. So the most important one is the specific soluble substance towards which the antibodies are produced. So this are demonstrated usually in the CSF, which can be identified with the help of precipitation technique. Next is the capsular palosaccharide, can be identified with the help of counter immunoelectrophoresis, and the antibodies against the other components of the organism can be identified via agglutination, precipitation, and you can go for mouse protection test or bactericidal test. Apart from that, you can go with indirect hemagglutination, indirect fluorescent antibody technique, and radioimmunoassay. Coming to the prophylaxis, the organism is the one that is usually involved in the causation of the disease pneumonia, and type-specific immunity is usually observed. Type-specific immunity means the production of specific type of antibodies. So with the help of this specific type of antibodies, you can easily identify the organism and can induce immunity by using vaccination. So here we have a complete polyvalent vaccine that has been produced in vitro to protect against this particular disease, but there is difficulty in protecting against 90 different serotypes of this organism. This polyvalent vaccine can protect against 23 serotypes, thereby giving a protection to up to 80 to 90% against this pneumococcal disease. And this vaccination can be given only to the risk individual. It cannot be given to any person, but only to the risk individual. Now, what type of risk individual do we have? We have the person who have immunocompromised state, where the person is where the lowering of the immune system is seen, or you have the person who have been suffering from malignancies, splenectomy, or sickle cell anemia. In those cases, this polyvalent vaccine can be administered, and the administration of the vaccine cannot be given to the children who are less than 2 years. And the treatment, as already discussed, these organisms are highly sensitive to different types of antibiotics. So initially the treatment was with the help of parenteral penicillin, but later we have found that the organism has developed resistance due to the penicillin binding protein modifications receptors. So here we can go for an alternative medicine or the antibiotic, which is amoxicillin, and for the resistant strain we can go for the 3rd generation cephalosporins. Vancomycin is usually given to the patient who are suffering from the fatal disease of the pneumonia and who are with the life-threatening condition of this particular disease. So this is about the pneumococcus organism involved in pneumonia.